Okay. Good morning, everyone. Today's case is about a 22-year-old female admitted to Dhoot for the treatment of sickle cell crisis. Her hospital course was significant for marked hypoxemia, thought to be related to acute chest syndrome, for which she underwent exchange transfusion. We became involved on day 3 of her hospitalization when a stroke code was called for progressive decreased responsiveness, followed by left upper extremity weakness. The decreased responsiveness had been over a period of several hours, so she was not a candidate for any vascular intervention due to an unknown last known well. As part of her stroke workup, we obtained an MRI of her brain. This showed diffuse punctate infarct, sparing the cortex in a pattern known as the Starfield pattern. And then less appreciable, but we also had an SWI sequencing, which showed some punctate microhemorrhages in the same areas. It's not as obvious here, but in a similar patient with a similar condition, here you can see diffuse punctate microhemorrhages on the SWI sequencing affecting both the cerebrum and the cerebellum. So what our patient has is known as cerebral fat embolism or fat embolism syndrome. This predominantly affects patients with sickle cell disease, typically who come in with severe involvement, cardiopulmonary, and with evidence of bone marrow necrosis, including thrombocytopenia and high LDH. This is classically a triad, though patients don't have to have all three, of hypoxemia, which our patient had, neurologic abnormalities, which can be defined quite broadly, including focal deficits, but also nonspecific, like confusion, lethargy, and coma, which can make this quite difficult to diagnose. And then finally, petechial rash, which our patient did not have. There are two causes of fat embolism syndrome. The one that we're probably all familiar with is related to mechanical trauma. So classically a long bone fracture followed by a pulmonary embolus. This is not actually what is related to sickle cell disease. For this, the theory is a biochemical damage where, as you can see in this diagram, there's damage to the bone marrow that releases free fatty acids into the circulation, which can go to the heart, the lungs, or the blood vessels, ultimately affecting the brain. In this, it's thought that the free fatty acids themselves cause inflammation and endothelial damage, which also explains those punctate microbleeds. It's not thought that fat microemboli themselves are what cause this syndrome. The treatment is mostly supportive. So exchange transfusion removes the sickle disease cells from circulation, thus preventing them from causing further damage in the bone marrow. Blood transfusion is also an option, but felt to be less helpful because it doesn't have that same removal of the diseased blood cells. Because this is it has embolism in the name, the syndrome is kind of embolic, the question was raised whether this patient should be on any anticoagulation. So heparin actually can increase free fatty acids in the blood and therefore is contraindicated. Also, because of all of the microbleeds, there is a risk of any anticoagulation causing further damage. On that same note, there's no evidence for oral anticoagulation and no evidence for steroids. Our patient is still hospitalized, but she's actually recovering well and planning to discharge soon to acute rehab. Thank you.
No, Roshni, thank you. That was an amazing case and presentation too. All right, so here's the mouse. Here it is. Okay, no slides. So today's speaker is Dr. Ann Augustine. She's an assistant professor of neurology and chief of neurology at the Durham VA Hospital and medical director of the sleep medicine program and sleep lab at the Durham VA. She has an MD from Boston University and a residency at Boston Medical Center. That's not Boston City, is it? Yeah. Okay, good. For anyone from Boston, Boston City is a big deal. They had one of the most famous neurology services ever. Derek Denny Brown was the chairman of neurology, and all three Harvard, BU, and Tufts had services on this one neurology service. It was quite a place in its day. She did a fellowship in, of all things, multiple sclerosis at the Brigham, now known as the smaller MGH. and a fellowship at sleep medicine at Boston Medical Center. And I just told her it looks like she's taking a trip to the dark side because she's actually going to talk about how sleep, memory, and dementia work. Her title is While You Were Sleeping, for those of you who can still sleep, the Lymphatic System at Work. So Anne, thank you.
Actually, Will, can I move for a second? Is there a way that I can get the speaker view and then project the? Oh, okay.
You can leave it like that. Just make it a little bit.
Okay. So, so the reason I chose this topic to speak about was recently at the sleep con. So recently, the reason why I chose this topic was that recently at the sleep conference, the lymphatic system was something that was of a lot of interest during the conference, and it's a relatively new concept, and I thought I'd talk about it today. So, what is sleep? We all have an idea or concept of what sleep is. It's a reversible state of reduced responsiveness to external stimuli. And during this time, we have relative inactivity accompanied by a loss of consciousness. And then the question is, why do we sleep? And we have some idea of why we sleep when we don't get enough sleep. We experience the effects of sleep deprivation. We notice that our ability to learn, our memory, and our attention is impaired. We also know that sleep has an effect in terms of regulating pain and our mood. We also know that sleep is an active process in the sense that metabolism, growth, and hormonal release occur during sleep, and it also plays a role in immune regulation. But what we also know about sleep is that it may have a role in terms of clearing metabolic waste from the brain. And that potentially could have a role in the development of neurodegenerative disorders. So the brain does not have a traditional lymphatic system the way we see it in peripheral organs, but the brain is a very highly metabolic organ. So how is waste cleared from the brain? Previous thinking was that waste products were cleared by intra and extracellular processes, autophagy and protein ubiquination, or by modulating proteins so that it could be specifically transported across the blood-brain barrier. The CSF, which is produced by the choroid plexus, flows to the ventricles and basal cisterns and acts as sink for the waste products to diffuse through the brain. So just to review the traditional lymphatic system, and basically blood goes into the arteries and then goes through the vascular capillary network, where it winds up diffusing out as interstitial fluid and winds up going into these lymphatic capillaries, eventually moving on, picking up solutes into the lymph nodes, and then being moved on to larger lymphatic vessels into the vein. But in 2012, the ideas of how metabolic waste is cleared from the brain was changed by Macon Niedergaard, who is a Danish scientist at the University of Rochester. And actually, most of the literature I present here and most of the literature that you see is actually coming out of her lab. And so this was a very seminal paper that she put out describing the lymphatic system. And it's called lymphatic because it's an analog of the lymphatic system, but based on glial cells. So in some ways, this is not a new concept, but without modern technology, some of these ideas were harder to prove. But these ideas have been around for possibly, for decades or even longer. So the idea that CSF enters a periarterial space, mixes with interstitial fluid, and has a role in waste removal have been put forth in years past. However, as I mentioned, we just didn't have the technology to look at these things. Now with advances in imaging techniques, we're able to look at mouse models and also look at humans and sort of see how fluid is actually moving to the brain. In mouse models, they often, a lot of the studies will use 2-photon microscopy and fluorescence tracers to look at, say, in vivo tracking of CSF in the brain. And in humans, This technology is still being developed to use MRI imaging to look at fluid tracing. So there's, what we are familiar with gadolinium-based contrast agents. Sometimes they can be a little problematic because they can have adverse effects, and it also takes a long transit time for contrast agents to move through the perivascular space. But other newer technologies include diffuser tensor imaging along the perivascular space, diffusion prepared arterial spin labeling, which uses blood as its own tracer to evaluate the blood-brain barrier, and chemical exchange saturation transfer, which uses MR spec to detect intrinsic compounds at low concentrations. So how does the lymphatic system work? Like, what is that exactly? So essentially you have your network of vessels at the ventral aspect or the basal aspect of the brain here in the circle of Willis, and you have blood coming through, and you have a CSF within the cisterns here, and CSF moves from the ventricles and cisterns to the subarachnoid space. From there, perivascular spaces surround the penetrating arteries in the brain parenchyma, and the CSF moves via arterial pulsations. This fluid then exchanges with interstitial fluid and solutes, and then winds up exiting via efflux pathways, draining solutes and fluid to the perivenous spaces and meningeal lymphatic vessels. So this is another view, a closer view of the lymphatic system. So perivascular spaces are formed by the end feet processes of astrocytes. These astrocytic end feet delineate the perivascular space from the brain tissue, creating tunnel-like structures that enable fast fluid flow. The position of the blood vessel, which sits slightly off-center of an elliptical-shaped channel, helps minimize resistance and optimizes fluid velocity. And as these vessels narrow, the size of the astrocytic end feet also decreases as well too. So as I mentioned, astrocytes, in addition to creating these channels, they also help to regulate perivascular and interstitial flow. And they do this through their aquaporin 4 channels, which are localized through the astrocytic end feet that are anchored by the dystrophin-associated complex. It's been shown that loss of aquaporin 4 leads to decrease in lymphatic flow. And it also plays a role in interstitial cell volume by regulation of interstitial ions, such as potassium, which has an impact on cell volume and interstitial space. So sleep, we all know it's important, but it's important for other reasons beyond what I had mentioned earlier in terms of sleep deprivation. It's been found to have a link between sleep and lymphatic flow. And we know lymphatic flow or lymphatic function is increased during sleep. And the reason why this relates back to sleep and as dementia, as we mentioned, is because lack of sleep can contribute to a buildup of waste products. A lot of times patients with dementia will report difficulties and sleep disruption before the onset of dementia. And healthy individuals who have poor sleep, they report deficits in memory, concentration, and mood changes. We also know that patients who have a history of epilepsy or seizures, if they're sleep deprived, they may be more vulnerable to having seizures. And that potentially could all be because there is a buildup of waste products that's not being cleared in sleep. But however, sleep difficulties of dementia might not just be a side effect of the disorder, but might actually contribute to the disease process itself. So faulty clearance of these waste products leads to protein aggregation, such as amyloid beta and tau. This in turn winds up disrupting neuronal signaling and contributing to the development of neurodegenerative disorders. So this is a very similar paper that I'm going to go over next. This also came out of Niedergard's lab and was written by Xi and other people in the lab. And so basically they looked at mouse models to track fluid flow. So they looked at in vivo 2-photon imaging to look at CSF influx into the cortex of mice. They looked at awake, asleep, and anesthetized mice. And they used a small molecular weight CSF tracers that were injected into the cisterna magna. And they used electrocorticography and EMG were recorded to monitor brain activity. So here you have the mouse where they injected the tracers. So they first injected dextran, FITC dextran while the mouse was asleep. And you can see in this picture on the left where you have the penetrating arteries where the green is diffusing through the brain into the penetrating arteries. While the mouse was awake, they then injected Texas red dextrin, and that's the red that sort of appears kind of orange at the superficial cortex layers over here. And you can see in the graph to the right that over time that there was a greater evolution of this tracer during sleep versus when the mouse was awake. Again, they this time looked at awake versus anesthetized, because if a mouse is anesthetized, it's almost kind of, a similar sleep-like state. And so again, here the mouse was injected with FITC dextrin. This is sort of yellow in the awake. And again, it's very similar to before, where you see a lot of this tracer sitting in the superficial cortical layers. Then they did it to the anesthetized mouse in the red, and you can see that the red is diffusing throughout the brain. And again, in the graph here, you can see the percent area covered by the tracers when the mouse was asleep or anesthetized versus awake. So why is there a reduction in CSF influx during wakefulness? It's likely not due to diurnal variation or arterial pulse waves. So we would think that during wakefulness, our blood pressure would be higher, therefore that would propel CSF influx. But using real-time iontophoresis method, which is a way of looking at extracellular space and volume, there was a reduction in interstitial fluid space during wakefulness. In anesthetized mice, interstitial fluid space volume increased by 60%. So it's felt that the flow of lymphatic fluid increases during sleep because the space between the cells expands, pushing fluid through the tissue. So about the role of norepinephrine. We know that noradrenergic signaling from the locus coeruleus contributes to cortical arousal. And they did another experiment using adrenergic antagonists which wound up increasing CSF tracer influx. So again, they used FITC dextrin, which was yellow, and while the mouse was awake, and you can see that the tracer sort of sits again in the superficial cortical layers. Then they used a norepinephrine, a receptor antagonist, which they use as a tracer. And you can see that when they did that to the awake mice, then you had more influx of the tracer into the penetrating arteries. They also did another study within the study. They also looked at the rate of amyloid beta clearance in sleep. And what they found was that, so they basically took radio-labeled amyloid beta. And what they found that amyloid beta was cleared 2 times faster in sleeping mice versus in awake mice. And they found that there was no difference in clearance between sleeping and anesthetized mice. So is lymphatic clearance circadian? So what's interesting is when you read these papers, just as in 2013 and 2015, which really wasn't that long ago, both of them asserted that it's not circadian rhythm, but rather the sleep-wake state itself that determines the volume of the interstitial space. But later research has shown that there may be a circadian pattern to the way the lymphatic system works. So in this case, they gave tracer to anesthetized mice at midday and midnight. And one thing you should remember is in terms of timing is mice are nocturnal. So they sleep during the day and they're awake at night. And so they found that 53% more CSF tracer influx went to the brain during the day compared with night in vivo along the middle cerebral artery. And you can see here in the right graph where they actually looked at 6 different time points and they followed the tracer intake. And there was an increase sort of during the midday when they would be asleep. And this suggests that there was, based on this curve, a circadian pattern to the uptake of the tracer. So then the next question they had was, is there diurnal variation into interstitial fluid clearance? And again, they injected a tracer into the brain, and they basically looked to see if they would find that tracer into the femoral vein. And they found, again, during the day, the tracer was able to move through and appear into the femoral vein versus at night that there was no flow of the tracer. So their conclusion was that clearance of solutes from the brain is upregulated during the day. So is there a circadian effect on aquaporin 4 localization? And again, using a tracer method, they found, or rather, I think there was radio labeled rather than tracer. You can see here on the left picture, there's aquaporin 4, and then lectin. And when they did the radio-labeled aquaporin 4, the white areas here during the daytime represent the aquaporin 4 being upregulated during the day, whereas at night, you didn't see that occurring. And then after exposing the mouse to constant light for 10 days, again, aquaporin 4, and so this is, they were trying to show that there was a circadian effect, was visible at rest versus not when it was active. So having constant light made no difference. So circadian control of lymphatic drainage. They also looked at to see, was there an effective lymphatic drainage? So what you need to know is, while CSF influx is increased during the day, lymphatic drainage is actually increased during wakefulness. That was nighttime for the mice. And they found that filling of the submandibular lymph nodes was 46% lower during the day compared with the night, meaning that when the mouse was asleep, lymph node drainage was lower. But again, after 10 days of constant light, that effect still persisted. So meaning that this was not necessarily due to exposure to light or darkness. So they've also, as I mentioned, that lymphatic influx is correlated with sleep, but is it just any type of sleep? So what they found is that it's actually found to have a connection with slow wave sleep or delta wave sleep. So here's a normal hypnogram. And this is what you would sort of see in a textbook, healthy 20-something-year-old person. And most of our slow wave sleep typically happens during the first part of the night in the red circles. And in the green areas here, that's our REM sleep, where we see most of it happening during the second half of the night. And anesthetics such as xylazine and dexmedetomidine, also known as Precedex, they're both alpha-2 adrenergic agonists, which cause delta waves in terms of sleep. Other anesthetics that are used in some of these experiments, such as isoflurane and pentobarbital, you see less slow wave components. And this is important when you read the studies, because some of the studies use different types of anesthetics when they're analyzing mice and CSF influx. And there are differences in their studies where they didn't achieve the same results, maybe due to the type of anesthetic that was used. So in this experiment, they looked at anesthetics and CSF tracer distribution. They tried 6 different types of anesthetics: ketamine with xylazine, isoflurane with dexmedetomidine, pentobarbital, alpha chlorelose, avertin, and isoflurane. And what you can see from this is that with the ketamine, xylazine, plus the isoflurane, dexmedetomidine, you saw the biggest amount of tracer uptake. Pentobarbital was kind of intermediate, but you didn't see a lot of uptake with the other types of anesthetics. And particularly when you looked at isoflurane, dexmedetomidine plus in comparison to isoflurane, there was definitely a difference when dexmedetomidine was included. So they then looked at correlation with delta waves. Ketamine with xylazine had the highest delta power, and isoflurane with dexmedetomidine had higher delta power when compared with isoflurane alone. We also next looked at heart rate and lymphatic influx. And ketamine with xylazine had the greatest tracer influx, but had the lowest mean heart rate. And that was also seen with dexmedetomidine having a lower heart rate when compared to the other anesthetics. So how does this all work with lymphatic system and neurodegeneration? And again, this is just another schematic of CSF flowing in through the perivascular spaces and picking up solutes and diffusing through. And these green bunches here represents amyloid beta plaques. And the idea is that as these amyloid beta plaques collect or accumulate, this winds up causing a reactive gliosis in astrocytes and microglia, which then contribute to even further slowing of lymphatic influx and further accumulation of the amyloid beta plaques. So this is a illustration of what sort of the proposed idea of what might be happening when we're having protein aggregation. So when they've looked at how amyloid beta, tau, and alpha-synuclein accumulate, you can see that there's a certain pattern how it accumulates. And when they've done lymphatic CSF influx by imaging, you can see that there is also a similar pattern in terms of how CSF is distributed throughout the brain, sort of indicating that there may be a pattern associated with CSF influx and the accumulation of these proteins. And it's almost like a sort of feedback loop in some ways, where you have different things that in fact affect lymphatic function, such as aging, sleep quality, sleep apnea. They mentioned cardiovascular disorders that reduce your lymphatic function. You wind up developing aggregates, and that goes back to reducing your lymphatic function as again in turn. And as these aggregates build up, you wind up developing neurodegeneration and then dementia. So what's the effect on sleep architecture? As I mentioned that from one of the previous studies, that there is a correlation with lymphatic flow and slow wave sleep. And again, as I mentioned, in a younger person who has sleep, you kind of see better consolidation of sleep. And up front over here, you wind up seeing, slow wave sleep that's not interrupted. But in an older person, we see much more fragmented sleep, much more arousals. And as we get older, we kind of lose some of that slow wave sleep. It's often sort of taken, in the literature, they say, as you get older, maybe around the age of 30, our slow wave sleep winds up, or deep sleep winds up declining. But that might also mean that our lymphatic flow also winds up declining as well with the loss of deep sleep. So what's the role of aquaporin 4 to neurodegeneration? In aquaporin 4 null mice, soluble amyloid beta clearance is reduced by 55% compared to wild type. There was a loss of aquaporin 4 localization with worsening Alzheimer's pathology. In cognitively intact 85-year-old subjects, there was no loss of localization. So the increase in amyloid beta that was seen wound up resulting in a loss of the aquaporin 4 that tends to localize to the end feet. There's also been more recent research looking at single nucleotide polymorphisms in aquaporin 4, and some studies have shown that depending on the single nucleotide polymorphism that's present or missing, that might have an effect on sleep quality and amyloid beta burden. And this can also have an effect on aquaporin 4 expression and changes in slow wave sleep as well. So there's no known treatment for Alzheimer's, and they suggest that lifestyle modifications may help influence its course. So some things that have been suggested, such as diet, consuming fish oils, intermittent fasting, body position while sleeping, alcohol, and exercise, and what's the effect of chronic stress. So one study looked at fatty acids and lymphatic clearance. So they looked at N3 omega-3 polyunsaturated fatty acids. Those are associated with having a lower incidence of neurodegenerative disease. Mice with high levels of endogenous omega-3 polyunsaturated fatty acids, these are transgenic mice, what they called FAT1 mice, had increased lymphatic clearance. Amyloid beta was cleared more quickly in these mice compared with wild type mice. And you can see that in the illustration on the bottom where you see tracer uptake that relatively gets cleared. Whereas in the wild type mice, it takes slower uptake and more time to clear. Next, I did an intraventricular cerebroventricular injection of amyloid beta. And the transgenic FAT1 mice navigated maze tests better than wild type. So they seem to have more preserved spatial and memory abilities. The increased presence of amyloid beta in the CA3 region of wild type mice, but to a lesser extent in FAT1 mice. And there was increased neuronal loss noted in wild type mice after injection of amyloid beta compared with FAT1 mice. So it appears that amyloid beta, or rather that having the omega-3 unsaturated fats present was protective. Other factors that have been looked at are things like sleep position. It's been reported that dementia subjects spend more time on their back when they sleep. In a mouse model, lymphatic transport is most efficient in the right lateral sleeping position. And it's thought that maybe the effect of gravity and a restriction in venous drainage on the carotid veins might have a contribution to position and lymphatic clearance. Alcohol, we know that that has a negative effect in terms of neurodegeneration. High levels of alcohol increase GFAP expression and delocalize aquaporin 4. Low doses, on the other hand, somehow increase lymphatic clearance due to decreased GFAP expression. Exercise has been shown to increase lymphatic clearance and reduce amyloid beta accumulation, and there's higher aquaporin 4 expression. And chronic stress also has a negative impact with decreased lymphatic transport and loss of aquaporin 4 polarization. So as I mentioned, that this is a relatively new field of inquiry. There's still a lot of unanswered questions. So some of the questions that they pose are, what are the molecular medicines that inhibit CSF influx during wakefulness, beyond the aspect of norepinephrine and arousal? What's the effect of REM sleep on lymphatic function? And it's difficult to create a model of lymphatic fluid flow to study some of these things, but hopefully with newer technologies, we'll be able to do that. And they also, some of the other questions include, what's the effect of long-term remodeling of the space, permeability, and patency of the perivascular space? And what's the role of the lymphatic system in terms of other aspects of GI signaling, circulatory system, and immune surveillance? So in conclusion, our understanding of the lymphatic system continues to evolve and may have important implications in neurological disease. New imaging techniques. Sleep, particularly delta sleep, plays a key role in lymphatic function. Lymphatic function is under circadian control as opposed to being driven by changes in light cycle. Fluid transport depends on astrocytes with localization aquaporin 4 at the end feet. Lymphatic clearance decreases with age. This can lead to buildup of toxic solutes and protein aggregates such as amyloid beta, and a decrease in aquaporin 4 expression, delocalization aquaporin 4 away from astroglial end feet, and reduced arterial pulsations lead to a reduction in amyloid beta clearance. And this buildup amyloid beta can lead to neurodegeneration. And so from Macon Niedergaard's paper, the most assured means of preserving lymphatic function is to just get a good night's sleep. So that's it. You're welcome.
Don't go away. So anyone with questions, just enter your name or your question in the chat and we'll call on you. Yep. Go ahead, Jill.
So I understand from Andrew that fasting increases Slowwave sleep. Anybody tried to prescribe diet or something and randomize them to medication and Slowwave sleep to see if we can target progression?
So can you repeat Jill's question just
Sorry.
because they can't hear it?
Oh, sorry. So Joel's question is whether, that mirtazapine increases slow wave sleep, and has anyone done a study to see if using mirtazapine in minimal cognitive impairment improves cognitive function or slows the progression of dementia? I am not aware of that study. Like I said, a lot of the papers that I've seen actually have just been sort of coming out in the last 3 years for the most part. And I think what they've looked at is what I showed earlier was the effect of anesthetics on sleep. But I think that was one of the things I was thinking about when I was researching this paper, like what other drugs could we be looking at and see if this makes a difference? And the question is also, mirtazapine, and Andrew can maybe answer this question. While, we see, there's what we sort of see in research and what we see practically. I don't know that I've always seen a lot of, this is what I thought about, I don't always see a lot of slow wave sleep in people. And it's kind of hard to control for, even in a laboratory setting.
So we'll call on the outsiders, but I just want to make a comment. So first, Ann, thank you. This is an important subject. It's certainly the flavor of the day. And the question is now, it clearly looks, this is the cause of Alzheimer's, but every 2 years there's another cause that doesn't pan out. And I think that's important to keep in mind. But this certainly is the flavor of the day. And we haven't heard a thing about it. But for the residents, what's important, what they're talking about here is, if you look at each cardiac cycle, almost 50% of the water that goes through your brain springs out through the endothelial cells into the brain, like one of those hoses that the kids play on in the middle of the summer. And that fluid, some of it is resorbed back, but a lot of it just percolates through the brain, clearing it out, and eventually has to go somewhere. And most of it ends up in the cerebrospinal fluid. But there's a barrier between the interstitial fluid and the cerebrospinal fluid, and that's these glial cells full of aquaporin 4. So, your idea, my idea initially about cerebrospinal fluid being 150 cc's a day, that's a net, but the entire churn of fluid is gigantic. And that's what's really important to remember in this system. Okay, so Anne, my question is the impact of systole on this system. If you ever see the heart contract, the brain expands. I mean, it's a traumatic event each cardiac cycle, and that has to have some effect on, squeezing things out like wringing a sponge.
So from what I read, I mean, so they did look at things like, I don't know the specific answer to that, I guess is my answer. I mean, when they had looked at, wakefulness versus sleep, what they found was that based on the idea that the heart is, contractile and would be, sending a pulse, that they didn't find, and that would be higher during wakefulness, they didn't find that there was increased CSF influx with that. So I don't know how that compares or interacts with the other mechanisms that are at play with the lymphatic system.
More to learn. And we'll start calling on things. I just have one more thing to say, because this is important. Every study in the world except one has said lack of sleep leads to dementia. But every study that has shown that association is biased by lag time. In other words, they're looking 2 or 3, 4 years before someone's got dementia, which means they already have dementia, and that's why they can't sleep. There's a single study that looked at sleep patterns 20, 30 years before dementia, and the effect was really teeny weeny. I'll send that out afterwards to people. So Marvin, do you want to go?
Yeah, I noticed her use mention carotid veins. I need to know about this. Is there a new name for the veins, or is that a mistake?
Carotid veins.
Did I say carotid?
Marvin, she didn't mean it.
Yeah, I meant it. I'll have to go back and look.
Corey,
Okay.
vasovasorum, whatever that is.
This is not something new I need to learn, right?
No, I don't think it is.
Okay, thanks.
How do you sleep, Marvin?
Great. And mostly on my right side too.
All right, I don't know who AJL96 is, but you have a question?
Yeah, it's me. Thanks.
Oh, Andy.
So there's a lot of literature over the past couple of years saying, sleep apnea that's not treated leads to the accumulation of A beta. And I was wondering if the lymphatics have any role in it.
I'm sorry, can you say that one more time?
Sleep apnea and dementia. So does sleep apnea lead to amyloid accumulation in the brain?
I think that's what's been proposed. I don't know about whether that's actually been. I think there's been some interest in that area, and I think that's what's been thought that might be part of the cause or process of amyloid beta accumulation.
So Ann, now that you're in the dementia field, the best policy is just to say, I don't believe anything that's out there and just move on.
Okay.
All right, a question from the audience. And if you just repeat the question.
Yeah, so I think the question was basically that if there is disrupted sleep and increased protein accumulation, does the sleep that you have afterwards, is that restorative and helps improve clearance? So as Dr. O'Brien said, we really don't know. No, I mean, really don't know. I mean, that's the thinking, right? That's if you sort of think about it from a sort of like a theoretical sense, they sort of write that, when we're sleep deprived, we wind up having like a rebound of slow wave sleep, and potentially that would wind up improving our clearance of those protein aggregates. But I don't think that anyone's actually done a study so far to show that that actually happens. But theoretically, that's what based on their models, that's potentially what should happen. So I think in one of the pictures that I showed, it was sort of showing how the protein aggregation sort of follows how the CSF tracer, when it was injected, it kind of starts kind of on the basal or ventral aspect of the brain and then kind of moves upward and forward. And that was kind of how the tracer goes. And I think that's what they were proposing in terms of protein aggregation.
All right, lots of questions coming in. Carmen, you're next.
Yeah, I was wondering whether, we see a lot of delirium in hospitalized patients. And as we know, the strongest predictors for delirium are sleep deprivation and immobility. And in fact, the anti-delirium is to turn the lights on, restore the day-night sleep cycles, and get patients moving. So do you know if anybody has studied the lymphatic situation among delirium patients, so we understand better the underlying pathophysiology of what exactly is making these people so loopy when they get sleep deprived in the hospital?
Yeah, I didn't see any research on that. A lot of the research that I did see is still very much in sort of like the basic science kind of area. And most of that's just come out in the past 3 years. And I think a lot of the stuff is still sort of being viewed in a very hypothetical manner and a skeptical manner. Well, I guess skeptical from Dr. O'Brien. But I mean, I kind of agree with him. There's a lot of buzz and a lot of interest in this topic. But, I don't know if it's going to be like the next vitamin D where everyone thinks it applies to everything and then maybe not so much. But to answer your question, I haven't really seen anything about lymphatic influx and patients with delirium.
Yeah, because this is the one population of people that when we get them to sleep and to move, it goes away. So you would think it'd be a great model to study.
Certainly.
Thanks, Carmen. Noreen, you're next.
Yeah, great talk. I was curious if there are differences due to gender and sex, and if that's been studied with particularly to age. I'm curious if there's a particular time of vulnerability where you're more at risk for developing neurodegeneration due to changes in your lymphatic system. Because in Alzheimer's, it's been studied that cholesterol, for example, at a certain age in their 40s may increase their risk later in time. And I'm just curious if a similar principle or concept applies in lymphatics. And then with regards to the gender, I'm just curious if the research in mice was done in a particular gender or if there's been any differences, if the sex differences has been studied in lymphatics.
I think those are really good questions. I think what you were asking about, was there any gender differences that have been seen in terms of lymphatic influx and development of dementia or neurodegeneration? And I have not seen any papers that commented on that, but that's an interesting idea to look at.
Do you know if the work in mice was done in male or female mice?
The paper, yeah, I was gonna say probably both. They didn't specify the gender of the mice or sex of the mice.
But don't worry, Noreen. Someone's looking into it, I'm sure. Let's see, more questions. No, Corey Myers, you had a question?
It's more of a commentary, just that this is a pretty long-standing concept that had been around and then dismissed and then now is revisited. So I was wondering, you were talking about sort of the landscape of dementia research and that there's something new and something old all the time and revisited, and many questions that remain sort of mysterious about it. But I was just going to say that, this has been around for 100 years, the lymphatic notion in the CNS, and then it was dismissed and then revisited. And so now it's popular, but it was popular once before. So it's really more of, what can we do to avoid bias that's going to limit our progress that has, this is an example of that happening in medicine.
Yeah, no, I agree. I mean, on one of my slides, I did point out that, these are not necessarily new concepts. I think what's new is our ability to be able to sort of look at these concepts now with using newer technology. And I think they're still trying to develop that technology to either prove or disprove whether the concept of a lymphatic influx or lymphatic function actually contributes to some of these issues around neurodegeneration.
And I would say, Corey, everyone always thought there was interstitial fluid, right? There had to be something around the neurons and it had to go somewhere. I think what's interesting about this was the idea that there are, at the end of the interstitial fluid, before it becomes CSF, there's a glial barrier to it, just otherwise it could have been just continuous and pouring in, and that there are things that regulate the rate of interstitial fluid clearance. And A beta, you know, everyone in this room is making over 1 billion molecules of A beta per second in their brain, 1 billion, and that's just normal. And that stuff has to go somewhere. So it's always been easy to imagine that if the clearance mechanisms break down, stuff will start accumulating. And but they just kind of brought more attention to it. So there's no doubt what they're studying is a real thing. The question is, does it really change under sleep and all these circumstances? Because remember, these are mice, and mice have been wrong pretty much 100% of the time in this field, or not wrong, but just not informative. Your turn. Say that mice have never been informative. All right, any other questions? So anyway, Ann, that was wonderful. Oh, you got one more maybe? Okay, Marvin, one more, and then we'll call it quits with the questions.
Yeah, is this one reason for dementia and hydrocephalus?
I'm sorry, say it one more time.
Is this why people with hydrocephalus get dementia, or NPH, why they get dementia?
I don't know. It could be one of the, it may have a contributing factor. I don't know that this has actually been fully researched, but it's their potential for that contribution of hydrocephalus contributing to dementia.
But there have been studies. I mean, whoever was the head of neurosurgery at Stanford did a very large study of putting shunts into people with Alzheimer's disease to increase CSF drainage to see if that had any effect, and it didn't. And almost everyone with NPH, they do have memory problems. Remember, NPH is a walking problem, but they all have underlying Alzheimer's and frontotemporal lobar degeneration. It's not that just the fluid makes them demented. All right, any more? No. Anyway, Ann, that was great. Huh? I know it is, but it's